Okay, hello everyone. I know everyone wants coffee on the last day. I'm Ezequiel. I'm a PhD student in Lab at the University of Massachusetts Medical School. I'd like to start by thanking the organizers for selecting my abstract about kinetic barcoding, which is this novel tool to estimate multi-temporal RNA biogenesis kinetics. So I'm pretty sure everyone in this audience knows how complex and multi-regulated RNA biogenesis is, and the different events need to happen in a coordinated way in order for a new molecule to be made. Over the years, we learned a lot about these processes and how they can affect things like gene expression, but we still have some things to learn about the underlying coordination between them. And this is what we're trying to understand in the lab, and we use a kinetic approach for that, and we have different pipelines that can estimate rates in this biogenesis process. And we actually have this new cleavage rate pipeline that was just published, the manuscript that you can look for. And we will soon have something about initiation, not rates, but just initiation as a process. But for this talk, I'll put my focus on this new method to estimate transcription elongation rates. So most of the current approaches to do this right now are either imaging or sequencing, and work something like this. You first clear out the elongating RNA pol2 from the gene body using a drug, for example, DRB, that arrests the polymerase initiation. And then you take out the drug, so pol2 can now elongate from the transcription start site, and then you can track the distance traveled by pol2 in a given amount of time, whatever you want. So with imaging, you can do that, of course, in high resolution, but low throughput. And with sequencing, one of the things you can do is use a molecule, for example, 4SU, which is a uridine analog that gets incorporated into the newly synthesized RNA. And you can then enrich for the new RNAs and track the distance traveled by pol2. And this is, of course, because it's sequencing, is high throughput, but not so high resolution as imaging was. So what we want to do is not compatible with these methods. So we want increased resolution, meaning that we want to be able to measure at shorter time points. This is a problem right now because of yield. You just don't get enough RNA. And then also accuracy that I'll talk a bit in a few slides. And then we also want to measure short genes and fast rates, which is a problem because you don't have enough temporal resolution. So we go back to the same issue. And the two things I'm mostly interested about, because I'm a biologist, are to be able to measure differential pol2 dynamics in the same gene. So region-specific elongation rates. And to be able to, and I'm not going to talk about this, to be able to measure pol2 elongation wave speeds. So is the first pol2 that travels along the gene faster or slower than the ones that come after that? And the reason for which we can't do this right now, or why it's so difficult, is because these techniques look at different populations of cells. So this is why we designed one single experiment to measure transcription elongation rates. And this is how it works. You have your cells in DRB, so pol2 is arrested at initiation. You treat them for 3 hours, just a normal treatment. And then you take out the DRB and you put in a nucleotide analog, just like 4SU, the one I was mentioning about before. This is not 4SU, by the way, which is here in blue. Sometime after that, you put a second nucleotide analog in red. Sometime after that, a third nucleotide analog in green. So just to be clear, at this time point between T2 and T3, all three nucleotide analogs are in the cells at the same time. So if this works, ideally, we can do a nascent RNA enrichment and then short read sequencing and then classify or assign the reads or the fragments to the time points they were synthesized. So in this talk, I'll show the experimental strategy we're using, the computational challenges we are facing and the ones we faced, and that we actually did this in cells. Okay, so before I go into that, I just want to describe this technique called SLAM-seq very briefly. So the way this works is you incubate the RNA with this 4SU, this uridine analog. It gets incorporated into a nascent RNA, and then you can induce a nucleotide substitution. You can switch from U, the 4SU is a U, to C. So you get a U to C or a T to C substitution. And then you do a total RNA sequencing, and you can identify which are the new molecules because they'll have a T to C substitution. So we got a lot of inspiration from this and other techniques to do something similar, where the three nucleotide analogs we're using have to have particular characteristics. The first one is something we need to be able to pull down from specifically. And then the other two, we should be able to induce a nucleotide substitution to identify them. But of course, to distinguish between them, that nucleotide substitution has to be different. So for the chemistry people in the audience, if there's any, the molecules we're using are 5-EU or 5-ethylene that we think we can selectively biotinylate and pull down, but shouldn't get any nucleotide substitution. The second one is 6-SG or 6-thiogonine, a G analog that shouldn't get the biotinylation, but it should get the G2A substitution. And then finally, the same one I was mentioning before, 4-SU or 4-thiouridine that shouldn't get the biotinylation, but should get a T to C substitution. So pull down and different substitution types. So this is how the entire protocol looks like. You have your nascent RNA there, just total RNA at this point. You do this thiolylation with IAA. So the 6-SGs should get a G to A substitution. The 4-SU should get a T to C substitution. And then we can enrich pulling down specifically from 5-EU, then do short read sequencing, find the substitutions, and assign reads to the time points they were synthesized. So we actually had to check the chemistry here because some things could react with each other, and we just need to be able that we can actually do it. I'm not going to show those results because this is not a chemistry conference, and I'm also not a chemist. But so what we need is for 5-EU to be able to be specifically biotinylated at pull down, and then we don't need any substitution, nucleotide substitutions in 5-EU. For 6-SG and 4-SU, we don't need them to react with the biotin, so we don't want to pull them down, but we do need to be able to induce these substitutions. And we collaborated with a chemistry lab at UMass, Jonathan Watt's lab, and Adam in particular did a lot of mass spec results that I do have if anyone is curious. So these conditions are actually very well satisfied, which was great. So at this point, we have the chemical confidence that we can actually do it. And I just want to go back briefly to this idea that we just don't have enough resolution right now to do these to satisfy our goals. And the main reason for that is that when people do not this experiment, but something that looks like this, they use 5-minute time points because that's basically the lowest you can go. So what we thought was maybe we could zoom in and just look at the very first few minutes from when taking out the drug, when Paul 2 starts transcribing. So what we did was to use 1-minute time points. So this is 1-minute resolution where we introduce each nucleotide analog every minute. And that means that the molecule we're pulling down from 5-EU is a total of 3 minutes in the cells, which is more than enough to pull it down. And the harvesting accuracy, which is basically the time between the last minute and the time you require to get the cells in a tube, is normally really high or just like a minute. But in this case, all the time points will be equally affected by that. That's different from the other methods where you need different plates of cells. So each plate will be affected differently by this harvesting accuracy. So we still need to sequence these, but we can, this is a dot plot for RNA. And we can already detect 5-EU after just a minute in the RNA, and 6-SG and 4-SU also just after a minute of labeling. So we are really excited about this, and I'll sequence it when I'm back. So the challenge here is that we don't really know if we can analyze data where we are inducing 2 nucleotide substitutions in the reads, right? Especially for mismatches and multiple alignments and things like that. So what I did was to simulate mRNAs for 100 genes with different elongation rates, because elongation rates is at the end of the day what we want to calculate. And we can also assign variable elongation rates to different features in those genes. And in this case, we define features as introns or exons, just to make it easier. So this is how the reads look like. And the color, each line is a read, not a read, sorry, an mRNA. And the colors represent the nucleotides, right? So if you see green, that means that read was synthesized in the last portion of the experiment. But if you see blue, this is only 5-EU, for example. So then I chopped the reads and did site selection like you normally do on a library prep protocol. This is now how coverage looks like. Now these are reads, not mRNAs anymore. And then mapping, right? And mapping could be a challenge because, as I was saying, you could have multiple substitutions in the same read. So the way we do this is using HiSat3N, which is a mapper that doesn't penalize for whatever substitution you specify to ignore. So if it sees a G2A between the read and the genome, it just doesn't care about it. And what I do is to map in parallel, allowing for G2As or T2Cs, and then merge those results and demultiplex. So this is how the alignment works. Most of the reads are uniquely aligned, higher than 95%. And then because of this, we thought, well, maybe we can assign the reads to the time point they were synthesized or simulated to be synthesized on. And that's the one on the top. And what you can see is that we can actually accurately assign the reads to the correct time points. And then because of this, we thought, well, maybe we can also estimate the simulated elongation rates we simulated up here. And we can actually do that pretty well. And as you can see, the first minute, the first time point is not as good as the other ones. And we think we can correct for that. Basically, what the problem we're having here is that some of the 6SGs or 4SUs that are there are not getting a nucleotide substitution. So we're just misclassifying some reads. But we will account for that, maybe using softwares like GrandR, which is actually the next talk. Okay, so we can do this chemically, we can analyze the data computationally. How does it look in cells? So I did 3 replicates of kinetic barcoding, and as controls, I used the methods that are already validated, that are either 5EU alone or 4SU alone. And as you can see, I have to use 5-minute time points here. I can't use 1 minute anymore, because these 2 techniques can only, like the lowest they can go is 5 minutes. So I had to do this. And as you can see, the other thing I didn't mention is that for 5EU, you only need 1 place of cells and 1 library. And for the other ones, you need 3 if you have 3 time points, and therefore 3 libraries. So this is actually way cheaper than doing just 4SU or 5EU. And I did short read sequencing on this. And then for each treatment, I calculated the elongation rates and found first that the treatments are similar between each other, and that the ranges or the means for elongation rates are really similar, not only for 5EU and 4SU and kinetic barcoding, but also other methods that have been widely used. And none of these are in HepG2 cells, which is the cell line I was using, by the way. We can also do a PCA for elongation rates. And we found that when looking at PC1, reads are clustering, or elongation rates are clustering by time point and not by sequencing technique, which was really good. So one of the things I want to briefly mention in the last few minutes is that this idea that we want to calculate variable elongation rates within the gene body, right? Different places in the gene that have different elongation rates. So there are some challenges we're facing with this. And the first one is that if we want to get feature-specific elongation rates, first we need to define the feature. And this is not trivial at all. And if anyone has any ideas about how to do it efficiently, I will gladly accept them. The other problem is that we probably are not going to be able to measure really short features. And then for the sequencing people, we also have an edge effect at the limits of the features that comes from fragmentation and site selection. But we do have this simple alternative that I'll show that it's not perfect, but it is something we have, and it's super easy to calculate. So if you think about it, this is how my data looks like. You have coverage on the y-axis and distance from the transcription start site on the x-axis. So at the first time point, you'll get a distance in blue here, D1. And then because just time passes by, Pol2 keeps traveling along the gene, and you'll find that when you introduce the last nucleotide analog, the distance is downstream from the first one, and then the third one downstream from the second one. It's just Pol2 traveling along the gene. So maybe what we can do is, and again, because this is in the same population of cells, maybe we can calculate elongation rates between these distances at every time point. So I brought 3 examples of how this would look like. This is real data, by the way. So we have a gene here where the first region, this one over here, elongation rate there is slower than in this region. So as Pol2 travels along that gene, at least in these regions, elongation rate goes up. This gene, the elongation rate doesn't change much. There are a lot of genes that look like this. And then this other one, elongation rate actually goes down when Pol2 travels along the gene. So the next step is clearly, well, to improve this metric, but also to be able to distinguish what are the features that might be driving these elongation rate changes. So what I showed today is that we can use 3 nucleoside analogs to perform a sequential labeling of RNA in one single experiment that we can have still to be sequenced, but we can have 1 minute resolution, that we have this computational workflow that can estimate elongation rates, that the average elongation rates from this technique, kinetic barcoding, can recapitulate published ranges, and that we're starting to analyze variable elongation rates across the gene body. So with that, I'd like to thank the collaborators from the Watts lab that did all the chemistry about this, my thesis committee, and my lab. They are awesome, and we're actually looking for experimental or computational postdocs. So if you like mRNA biogenesis and maybe you like dinosaurs too, just join us. Yeah, we're fun. Yeah, thank you.
Thanks. Cool stuff. I'm curious. So when you do these modifications to pull down, do you know or have any idea how much you're affecting the RNA structure and thereby what you're actually measuring?
Yeah, we might hear about that in the next talk. I'm not sure, but we are not, in principle, affecting. So we check viability. No issues there, because especially these are so short that it doesn't matter. We didn't check secondary structure. Maybe we should, but yeah, definitely. Mm-hmm.
Okay, I want to ask something.
Mm-hmm.
So you're doing it for 1 minute, so I can only assume you don't put it back in the incubator and
No.
then take it out.
I know.
And so is temperature a thing in elongation? Because yours was 2 kb per minute,
Yeah.
and you showed that it's kind of similar.
Yeah.
So
yeah, I'm not putting it back. And I do agree with you. Yes, probably. So I have this weird idea where I want to bring the water bath into the hood and then do it there. Yeah.
okay, maybe last one.
I also have a technical question. I was wondering whether on take of the chemical analogs and their incorporation rates are different between the analogs, and whether you somehow use this in your normalizations.
Yes, right. They are different. Not only the incorporation rates, but the substitution rates are also different. So those are the results I didn't show. We actually estimated those. We estimated the IAA treatment, how efficient that is with mass spec, and then we did Sanger sequencing to calculate what's the substitution rate. But the substitution rate is also context dependent. So we're working on like a library of context for the nucleotide, the modified nucleotides to account for that in the pipeline. Yeah. Thank you.
You take out DRB. What's the kinetics of that compared to incorporation rates?
Okay.
So,
right.
I mean, you have to consider that, and also there has to be a burst the first round,
Yes.
and then the reinitiation. And do you see any evidence?
Yeah, we didn't look for it. We should, but I watched many times DRB. Maybe it's not enough, especially for the one that's in the cells, probably. But we are seeing RNA. So we think it's, I mean, something is being synthesized. Yeah, we might need to be able to measure better if DRB, how much DRB is still in there.